Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is September 9th, 2024, and we're talking with Marina Silveira, who is assistant professor in the Department of Neuroscience, Developmental, and Regenerative Biology here at UTSA. Marina's been at UTSA for slightly less than a year, but has been busy, successfully set up her lab, and it's become a bustling, busy place just recently. Hi, Marina.
Hi, Charlie. I'm very happy to be here.
So Marina's work is on cells and circuits of the auditory system, especially the inferior colliculus.
Exactly.
Which is the largest of the brainstem auditory nuclei and the most accessible, as it sits right at the top of the brainstem at the level of the midbrain. Given that, you might expect it to be the most well-characterized of the auditory nuclei, but in fact it is not.
It is not.
And Marina is working to fix that.
Hopefully, that's the goal of our lab.
So how about getting us started by just telling us about the location of the the inferior colliculus in the auditory system, where it sits, and what we know about it in general terms.
Exactly.
And then we'll go on from what we don't know.
Exactly, yeah. So I'll start by telling you a very quick story of when I joined the auditory system as a researcher, right? I came from a different field. And when I joined, I went to this talk. It was more like an outreach talk, but it was like a talk about the auditory system. And the person who was giving the talk, she said, so hearing begins in the ear, and a bunch of stuff happens until you reach to the cortex. And I was like, wow, I was just starting working in inferior colliculus, right? Uh-huh. And this really opened my mind about, there is a lot that we don't know about not only the inferior colliculus, but also the auditory brainstem, just because for a long time the field was built on hearing being majority processed in the ear, like a jump to the cortex, and there's so much that happened in between that we are just now, for the past few decades, starting to understand that. So it's very important to realize that there's so much processing happens before you reach higher cortical areas. So here begins in the ear, as I said. So you have the cochlear nucleus as the first stop, the first synapses of the auditory nerve. From there,
So all the auditory nerve fibers
to go to the
synapses there,
in
right?
the cochlear nucleus, right? And then some of those cells from the cochlear nucleus, they can project directly to the inferior colliculus. Other cells, though, they first project to the other auditory nuclei in the auditory brainstem before it goes to the inferior colliculus. So we usually say that the majority of auditory information is processed in the inferior colliculus before it reaches thalamus and auditory cortex. So that's why we talk about the inferior colliculus as the hub of the central auditory pathway. And there's a few important things that when I'm trying to convince people that inferior colliculus really matter for auditory processing, right? So the first one of them, there's some studies showing that the inferior colliculus is extremely important for speech perception. So if the inferior colliculus is damaged, it becomes very difficult to understand the speech.
So is that just because you can't hear anymore?
You can still hear. So your cochlea, let's say your cochlea is intact, you can still hear, but you cannot understand what's being said. So the processing of the speech. And also sound localization, which the inferior colliculus also contributes to it. So neurons in other auditory brain regions are also important for sound localization, but neurons in the inferior colliculus are also important to detect where a sound is coming from. With that being said, there's so many neurons in the inferior colliculus. So it's estimated that the number of neurons in the inferior colliculus is 5 times more all the auditory brainstem combined. So a lot of auditory processing is happening in the inferior colliculus. So these neurons, they send projection to the auditory thalamus that then project to the auditory cortex, and then we have those descending cortical projections back to the inferior colliculus. So the whole auditory pathway is somehow integrated by these ascending and descending projections. And for a long time in our field, people refer to the inferior colliculus as this hub. So it's the hub of the central auditory system, it's the hub of the central auditory system. And all we knew about cell types was that, okay, so it's the hub, but how is auditory process, what are the mechanisms underlying auditory process in the inferior colliculus? So all we knew about cell types was like neurons in the inferior colliculus, they are either GABAergic or glutamatergic, estimated to be around 75% GABAergic, give or take, depending on certain species, and around 25% GABAergic neurons. We also knew that they have two types of morphology. So stellate, which are like the stellate-shaped cells, or the disc-shaped cells, which are more like more thin cells that kind of go diagonal in the inferior colliculus. And that they have a large variety of firing patterns. So meaning that when we look to their activity or we see how these neurons respond either to sound or to like a current step, the firing pattern of these neurons is very variable. And different than what we see in other brain regions, this firing pattern and the morphology, they don't correlate with each other. So when people try to identify cell types...
Just tell me what that means exactly, because there are really just two kinds of morphology you described, the sort of flat cells and stellate cells.
Exactly.
And then what would it mean for the variation in firing pattern align with that?
So if we were to recording from the cell, record the activity of the cell, it could have, for example, an adapting firing pattern. So it starts with like some action potentials and then adapts. Or a sustained firing pattern where it doesn't matter either how loud is the sound or how depolarizing is your current step, the cells keep continuous firing in a sustained way. Some cells are onset. They only fire one action potential in response to either a depolarized current or a sound presentation.
So we normally think when we see that kind of difference, that that's caused by differences in expression of ion channels on the membrane of the two cell types.
Exactly.
That doesn't necessarily imply a morphological difference, but we're used to cells that are morphologically similar to have similar ion channel expression patterns.
Exactly.
And so do you conclude that cells that look similar have different ion channel?
That's not the case in the inferior colliculus, because we can have cells that have the same morphology, but they have a different firing pattern. So it's a little bit different than what we're used to in other brain regions. So this was a limitation in our field, right? It's difficult to identify cell types. How do you understand how certain types of neurons contribute to sound processing, and also how these neuronal circuits are organized? We need to first of all, identify the cell types. And this is...
But how about natural stimuli? So if I give a current pulse into a cell and I see changes in its firing pattern, I usually attribute those to ion channel expression. But in a real life situation where a cell is listening to stimuli, sensory physiologists normally don't go first to injecting current. Normally they give some kind of natural stimulus, and they have 12 different cell types based on how they respond to sounds or something like that.
Exactly.
And what do you get? What do you get from that in the
inferior colliculus? So maybe let's take a step back and talk a little bit about the cochlear nucleus, where these responses to sound of the cochlear nucleus, they are so much more clean and well characterized. So the cochlear nucleus, we have these different cell types, and each one of them respond differently to auditory stimuli. So some of them, they respond with like an onset firing to certain auditory stimuli. Another cell type responds to a sustained fire to an auditory stimuli. And for a long time, and even nowadays, when we are doing what we call like in vivo experiments, when we have an electrode in the brain, in the cochlear nucleus, we are recording from a cell, present a sound. Based on the firing response, we can identify which cell we are recording from. So people have been trying to do that in the inferior colliculus, so presenting sounds and trying to identify how those cells respond to that certain sound. But the responses are so heterogeneous that this will not be enough to be able to classify those in cell types, if that makes sense.
Oh, so there's too many.
Too many.
Too many different kinds of responses.
Too many different kinds of
responses. The cells do pay attention to frequencies, so they have a preferred frequency. They have a tuning curve.
So they are in the tonotopic axis, right?
But beyond that, all bets are off.
Yeah, exactly. So it was for a long time very difficult to classify the cell types. And then, do you have something to say?
No.
You're gonna go okay.
No, keep going.
And then what happened? So as soon as I was starting my postdoc, I was working with Michael Roberts, who really was the person who pioneered how to identify cell types in the inferior colliculus. And Michael, he brought idea that has been used in other brain regions. That is to use neuropeptides as markers, so neuropeptide expression, as markers to identify different classes of neurons or different types of neurons. So that's how he started his lab back in 2016. So
in a way, this is like separating GABA and glutamate neurons, except there's just GABA and glutamate, and it's not enough. But different cell types express other co-transmitters, these short peptides, in the thalamus, in the cortex, and basal ganglia. These things have been super valuable at sorting out the differences among interneurons, especially.
Exactly. So that's what we bring in this expertise. Like I was learning from other brain regions, bringing this expertise to the inferior colliculus. Could we implement that and finally be able to identify different classes of neurons in the inferior colliculus?
What are the 5?
So we started with 2, right? We started with VIP, for vasointestinal active peptide, and neuropeptide Y for NPY. So how did we do that? So we used transgenic mouse lines in which we can visualize these neurons by the endogenous fluorescence of this genetic manipulation of this mouse. So let's start with VIP first. And we found something very interesting, that VIPs, neurons that express VIP in the inferior colliculus, they are glutamatergic. And this is different from anywhere else in the brain. And this was validated over and over again by different labs. So they're glutamatergic. So we found VIP to be a class of neurons that are glutamatergic. They have a stellate morphology, so as homogeneous. All the VIPs, or I should say almost all of them, I think it was 95%, had a stellate morphology, a sustained firing pattern. They're glutamatergic, and they project to different auditory nuclei and other no-auditory nuclei as well.
So I would conclude from that all that variation in firing pattern is meaningful.
It's meaningful, exactly.
And that eventually there may be 62 different firing patterns. There are going to be 62 different cell types.
Exactly, different cell types. And on the other hand, the neuropeptide Ys, they were GABAergic, as we have seen in other brain regions. They are a large proportion of the GABAergic neurons, so we usually refer to NPY as a class of GABAergic neurons. We are still in the process of subdividing them. We believe they might be subdivided in other cell types as well. But they're also stellate, they are GABAergic, and they have a similar sustained firing pattern. So these were the first 2 cell classes in the inferior colliculus.
So that means stellate cells aren't all GABAergic.
No, because VIPs are glutamatergic.
So there's not even a good correlation between GABA glutamate versus stellate.
Exactly, yeah. So it's very interesting. I usually think of inferior colliculus as I think for me it's so fascinating because it goes against all the rules from what has been described in other brain regions. So I think that's something really fascinating about inferior colliculus, and we're just now starting learning. So using... And so Nace Golding at UT Austin, like our neighbor here, he also found CCK to be a marker for a large population of disc-shaped glutamatergic neurons. And another group just published that somatostatin can also be used as a marker of a cell class of neurons in the inferior colliculus. So now as a field, we are beginning to understand what can be used as a marker to identify these cells. And we can finally use all these neuroscience tools to selectively target these neurons, begin to understand where they project to, how they contribute to local circuits in the inferior colliculus, how they affect their postsynaptic targets, either ascending or descending projections. Because one curious thing about the inferior colliculus, GABAergic neurons, they stand long range. inhibitory projections.
So just because they're GABA neurons doesn't mean that they stay local.
No, doesn't mean they're
and there are a bunch of different projections of the inferior colliculus, and we really don't know whether they all come from the same cell types or which each one of them has. Is that true?
Is that true? That's true, and that's still a big question. So one question that I always get when I'm presenting my work, so I usually talk about NPY neurons and the fact that we show that they can project locally, they can project contralateral, they can send long-range projections to the auditory thalamus. And
is this an either thing, or is that an and?
That's exactly the question. Is the same neuron projecting locally, contralateral, into the auditory thalamus? We don't know yet. We want to test this hypothesis. So the way that we would do that, for those of you who are not familiar with neuroscience techniques, a possible way is to use what we call retrograde tracing. So we can use 2 different colors of retrograde tracing and label 2 different portions of the brain. And because it's a retrograde tracing, will be retrograded, transported for the neuron that we want to test, whether it's projecting to this other nuclei. And then let's say we use green and red. And then we look back in the inferior colliculus. And if you have an NPY neuron that collabel with green and red, it means that NPY neurons send projection to these both targets. So it's what we call collaterals, right? Sending axons to both locations. But if it has only the red or only the green, it's probably only contributing to one nuclei rather than the other. So this is one experiment that, is on our to-do list. We just haven't done yet, but it would be very interesting to know how the circuits are organized and where this neuron projecting.
But right now, you could probably say what the VIP neurons, the neuropeptide Y, neuropeptide Y neurons do. Because you have a genetic marker, and you can look at where their axons go.
Exactly.
And as a group, what do they do? Are they interneurons? They project contralaterally? They go everywhere?
So as of now, we know they project locally. So they provide inhibitory inputs to local neurons in inferior colliculus. They also project to the contralateral AC, and they also project to the auditory thalamus as a cell type. So that's what we know so far. We have some preliminary data that projects to another nuclei, which is the nuclei of the breaking of the IC that connects the inferior colliculus to the auditory thalamus. So these are the projections that we have identified so far using this.
So this method, cell by cell type, will eventually answer all those questions. It's just a matter of having the right mouse. You need a mouse for this cell type and a mouse for that cell type. Because that's how we do it.
That's how we do it. So we are really, compared to this striatum, we are still a child identifying the cell types in the inferior colliculus.
I say, I can do anything if I have the right mouse.
Exactly, yeah. So we are still identifying which are the markers for cell types in the inferior colliculus. And then, after we do that, we can then give a next step and identify, in addition of how they contribute to the local circuits and ascending projections, also how these neurons respond to sound, right? Because as you said, the inferior colliculus is extremely accessible in the mouse brain. So when we're doing in vivo experiments, we can just put a pipette in there with no trouble because it's very shallow, right? It's on the surface. And we can use a technique that's like optogenetics, that we can, optotagging is how we call, that we can selective target our recordings to certain populations of neurons if we know which those markers are. So what we are trying to do right now is to record specifically from NPY neurons or from VIP neurons or from other populations of neurons that have been identified and see how this distinct class of neurons respond to sound. Is their frequency tuning similar in the same phenotypic organization? Is how they respond to AM sounds or FM sounds, complex sounds.
So there's a lot of different sounds you can give. I mean, how do you characterize a neuron?
It depends on your question, right? It depends on.
So if I'm looking for cell types, I mean, from what is known about inferior colliculus, because people have recorded responses before, they just never knew what cell type they had. So inferior colliculus neurons have any, specific to the inferior colliculus kind of stimulus that they're super sensitive to, or some dimension of the stimulus that is some. I mean, I'm thinking about what you said about speech sound.
Yeah.
And so if my inferior colliculus isn't required for me to hear, but is required for me to understand speech, then there must be something about the speech sound, that inferior colliculus, the kinds of sounds that are associated with speech.
That's a good question. So neurons inferior colliculus, they respond to amplitude modulated sounds. There is also a very interesting feature of the inferior colliculus that has been mostly characterized in bats, with some studies that came recently in mice as well. And from our lab, we have a study in progress about that. But from Michael's lab, actually, when I was a postdoc there, not from my lab here. But it's called frequency modulated sweeps. I don't know if you ever heard about it, but it's like very fast changes in frequency modulated sound. So it goes like upward sweeps go from lower frequency to higher frequency, or downward sweeps goes from down frequencies to lower frequencies.
Like the beginning of an Emerson, Lake Palmer song done on a synthesizer.
Yeah, like a very high, very... Exactly, that's it. And then what has been shown is that neurons, the inferior colliculus, some neurons, they respond best to upward sweeps. And some neurons respond best to downward sweeps. This is like really broad, and there's still a lot of work in progress on that. But the idea is that some neurons respond best to upward sweeps, and some neurons respond best to downward sweeps. And FM sweeps has been thought to be a feature that is important for speech processing.
So this is temporal.
Yeah.
Right? It's because they have exactly the same frequencies in them.
It's just changing the speed.
Not like what we normally think of the cochlear nucleus or someplace doing. Because those things are listening for either time differences or for frequency differences, or am I wrong about that?
There is like a new study though that showed that octopus cells, which are localized in the cochlear nucleus, they can detect the changes in FM sweeps as well. So that's where things get really complicated with the auditory system in general, because first of all, things are very connected, right? But also some neurons have similar ability to encode sound processing. So that's why, this is my opinion, but it's like some of the times gets so frustrating to silence or completely silence the auditory region, and you expect you're going to see a huge difference. The mouse will be completely deaf, but a lot of the studies that have been done, the mouse can hear just fine a lot of those times, right?
Yeah, so a lot of what you see may not be generated in the neuron that you're recording from, but actually be generated at some other level in the auditory system and then sent. And you can't just assume that at the bottom of the system, everything is where everything begins.
Yeah, exactly.
And that it ends up at the cortex.
Cortex, exactly, because a lot of this processing happens in between the ear and the cortex, right? So there's so of course that's how science works, but so many labs are focused on your just distinctive nuclei. Right? But how is everything connected, right? So I'm studying a very niche in the inferior colliculus, right? This population of neurons, how they respond to sound, what kind of ion channels they express, where they project to. But you always have to step back to the big picture, right? How are they combined with the rest of the auditory pathway to then sound processing allow us to make sense of sound? So everything's largely interconnected in the auditory system.
So does that explain, the most puzzling thing you said was that if you make a lesion in the inferior colliculus, you can hear, but you can't understand speech. It seems to me that all auditory projections have to make it through the inferior colliculus somehow in order to make it to the cortex.
Yeah.
So is the cortex required for auditory perception? If I didn't have an auditory cortex, would I describe myself as deaf?
That's a good question. And there are some studies showing that when you silence auditory cortex, mice at least can still hear well when do the auditory tasks that they were doing before. And I think this has been done even pretty early in the days. But that being said, it's difficult to know whether when you're silence a brain image, if you're silence everything, which are the limitations of this technique. So it's hard to know, like, I think it's a very strong word to say it is required, it's not required.
Uh-huh.
So
it is a strong word. That's
it is
why
a
it's so
strong word.
powerful.
Yeah, so I would not feel comfortable saying it's not required or it's required. There's certainly studies moving towards that and silencing different regions and see how they can, how this can affect the auditory process.
So somebody is described like as having central deafness.
Mm-hmm.
Does that usually mean somewhere in the cochlear nucleus or something like that? Is that what it usually means? Or does that usually mean something like a lesion in the cortex, like a stroke?
Yeah. I think I'm not sure if we know for sure, like when you say central, it can be related to either auditory nuclei.
Uh-huh. So we don't pin it down.
I'm not familiar with that.
If it's not in the ear, it's central.
Yeah.
That's the definition of central.
Yeah, I would think so. Yeah, I would think so. When you do like ABRs, auditory brain, you can see like depending on the peak of the ABR, you can see where is auditory brainstem responses that you use to check how well is your ear. So it has like the peaks and the waves of the ABR. Each one of those will correspond to a certain area in the brain.
This is a cool feature of the auditory system that other sensory systems don't really have, that you can detect with as a field potential.
Yeah.
The components that come from different levels of the
exactly.
system.
And you can estimate how well.
So there's nothing like that in the visual system.
That's true. That's true. Yeah, the auditory system it's very fascinating, and for me, like what's very fascinating about the speed, right? The speed about how these inputs are computed in the brain, right? So I'm just talking to you, and you're nodding because you can understand simultaneously what I'm saying, and then you respond back. So this is like the microsecond time scale, right? And it's so fast and goes for so many stops. And you can still, like, the fast of it for me is what's the most fascinating.
It's all connected in loops. It's not actually that surprising we don't understand it.
Exactly, yeah.
So I wanted to ask you about get back to cell types a little bit in inferior colliculus. So one of the things that happens in other places, happening in other places right now, is that people are doing these large-scale transcriptomic studies to try to define cell types according to expression of lots of different genes, coordinated expression of a lot of different genes that create clusters in this multidimensional gene space. And you don't necessarily worry about what each of the individual genes is, but you can point at a cluster and you can say, I think this is probably a cell type.
Yeah.
And in those, that becomes really useful when you have a marker for a cell type and you can say, that cluster has that marker. That one is that cell type. And then you can look at all the other things that are being expressed in that one and find a bunch more markers for that cell type. And it sounds like you'd love to be able to do that because you're a little short on markers at this point for cell types. So is there anything being done like
there
that?
Is. So there is like there's two labs, one lab working on it that I'm aware of. They are doing RNA-seq to identify like all the markers, possible markers, and would be like a huge contribution to the field when that's done. Because then, as you said, we can even like subdivide the marks that we have now to get to more like homogeneous cell types. And then the challenge, of course, comes to make mice that have this marker so we can then study the features of these neurons, study the electrophysiological properties, their anatomical properties, and really validate whether they are a marker for a cell type, right? If they're homogeneous in their properties.
Yeah, we act like these mice come out of nowhere, like that they're easy. Like a mouse that expresses some reporter under the control of the neuropeptide Y.
Yeah, so
But you can't just look up in a catalog, I've just discovered this gene and I want a mouse that's going to mark that.
Yeah, you would have to get this mouse made, which is about like $20,000. $20,000 you can get a mouse done.
And it takes time too.
It takes about 6 months. 6 months and $20,000.
Although it's miraculous actually that it's that cheap and that fast. It used to take a lot longer. Yeah,
so now it's in, but like for the most like widely expressed markers like neuropeptide Y, VIP, we have mouse available in a catalog, really. that we can just go there and select it. But when you get to a point where you identify new markers, it can get a little challenging unless these are the same markers that have been studied in other brain regions.
So up to a point, you can just sort of look up what markers have been useful other places. Until you exhaust that list, you might make a whole lot of discoveries.
I make a lot of those.
And it's a pretty long list.
Yeah.
It doesn't go on forever. Probably not enough to cover all the cell types that you're looking for. But
Exactly.
But you probably find a bunch of them. Do you have favorites? Want to try next?
I've been thinking about CCK. CCK has already been described in inferior colliculus, but then we have somatostatin. And we know that PV doesn't seem to be a marker for a cell type in inferior colliculus because they are a little bit not as homogeneous for the PV. But there are other neuropeptides that are expressed in the inferior colliculus as well. So we have one thing that we found in our lab while I was in Michael, and we're continuing here in my lab, that neurons that have the receptor for NPY, they're pretty much almost 100% of the glutamatergic cells, which is definitely not a cell type or a class of neurons, but it's still very interesting that our glutamatergic neurons have the same receptor, but the GABAergics don't have this receptor. So not a cell type or a cell class, but definitely like a common feature across all the other glutamatergic neurons. And like in cortex, for example, they use serotonergic receptors to identify cell types. We know the serotonergic receptors are expressed in inferior colliculus as well. So we are working on identifying whether they're by a homogeneous group or not, or if they're identified by different classes of neurons. There are also dopaminergic receptors in inferior colliculus. I don't know if you know that, but the D2Rs are in inferior colliculus. As far as we can tell, and again, I think it's so hard to say negative data, right? But as far as we know, there's no D1 receptor. So only the D2Rs are expressed in inferior colliculus. So
these are convenient ones because there are mice.
Yeah.
There are mice for those.
There are mice for those. So this is like probably the place to start. But hopefully the RNA seq will come soon, and we'll have more tools to identify other cell types in inferior colliculus.
Great. It looks like the path is all laid out for you. Is that the way you feel?
Yeah,
I just have to do it now.
Yeah, for sure. We are mostly like in our lab right now, we are really looking into neuromodulators and how they change the activity of neurons in inferior colliculus. Because it's a whole other story. A lot of neuromodulatory inputs arrive in inferior colliculus. Serotonin, dopamine, NPY, and nobody knows what it's doing and how it's contributing to auditory processing. So this is a lot of our focus now, identify like the function of these neuromodulators in inferior colliculus.
So that's a whole other thing, is once I found out this peptide is present,
what is it?
I have to figure out what it does.
That's exactly what we're doing now. And this is like a personal interest of mine too, like, because I came from the hypothalamus, where all these neuromodulators matter so much for reproduction, control of metabolism. So I have always been really curious about what they're doing in the sensory of auditory processing.
Great. Well, thanks a lot.
Yeah, thank you.
Marina.
That was great.
That was really interesting. And this has been Neuroscientists Talk Shop.
Thank you.